Microsatellite markers. Repeat 1 to 7 base pairs, with allele size 100 to 400 base pairs, such as STR marker, SSR marker, and ISSR marker, as mentioned inside slide and next slide. The steps of fluorescent microsatellite markers. Initiate by PCR amplification, using fluorescently labeled primer, and mix product with size marker, and separate product by capillary electrophoresis, to detect larger and smaller allelic fragments, and detect heterozygous and homozygous alleles as well, as mentioned in next slide. Microsatellite markers have 4 types: perfect microsatellite markers, imperfect microsatellite markers, compound microsatellite markers, and complex microsatellite markers, as mentioned inside slide. Continuously. Mutation rate of microsatellite markers. Range between 0.001 to 0.000001 per locus per generation, as mentioned inside slide. In comparison of microsatellite markers and SNP markers, microsatellite markers depend upon length differences, while SNP markers depend upon one specific different, as mentioned inside slide. On the other hand, mini-satellite markers repeat 15 to 35 base pairs, with allele size 400 to 1000 base pairs, such as VNTR markers, as mentioned inside slide. So, micro and mini satellite markers depend upon southern blot technique by hybridization with probe to detect target sequence, and PCR with paired primers to flank variable sequence. Therefore, micro and mini satellite markers consider co-dominant nature markers with highly polymorphic loci, with less susceptible to DNA degradation, to study closely related species. Hence, micro and mini satellite markers use in population studies to find differences in alleles frequencies, to trace specific alleles through pedigree, to identify specific genes in chromosome, for forensic studies.